So we're now going to switch a little bit back the other way to talk about what's going on and what's coming. We're going to have another speaker from the University of Oxford who's been researching this area, and of course what we're all interested is where the treatment's going and where that will take us. And it's my pleasure now to introduce Professor Beth Psaila, who's going to tell us about MPN targeted immune therapies, which is where we may be going in the future. Thank you, Beth.
Thank you, and just to add my thanks as well to MPN Voice and to Claire Harrison in particular for organising this day. I know from talking to my patients over the last couple of months, they've been so excited about the opportunity to hear from all the speakers today, so thank you for all the hard work that goes on. So I'm going to be talking a little bit about trials that are coming. So actually we haven't got any patients enrolled in these trials yet in the UK, but we're about to change that, I think, next week, which is very exciting. Some of these trials are already open in the US and elsewhere internationally. And what we're talking about really is using a type of treatment called immunotherapies, which are basically agents given to people to try and arm their own immune system, to wake up their own immune system, to target specifically the cancer cells or the MPN cells for this patient group. So it's quite a new type of therapy that hasn't really been used in this setting before, but is already used in other cancer types and other disease settings. So the idea is that basically patients with cancer have cancer cells. Obviously, you use immunotherapies that come in different flavours, and the idea is you're able to clear these cells then from the system. The types of immunotherapy I was going to talk about today are really these two shown here. So cancer vaccines, and I'll come on in more detail in a second to talk about how those work and what the aim is with those, and then therapeutic antibodies. So vaccines, we all know about vaccines. We've all recently been through the COVID vaccines and flu vaccines and everything else. They're actually quite successful already in the clinic in the context of patients with melanoma, so skin cancers. And then antibody therapies, again, in other settings, they're hugely successful and have really revolutionised treatment for patients with leukemias, with myeloma, and with lymphomas. So they're not new therapies as a whole in the context of cancer treatment, but they are very new to MPNs. And I won't really talk about T-cell therapies at the moment because that's still in years to come, I think, in this setting. And just to say that we're very familiar as a field with using agents that arm our immune system to attack the cancer. For example, we all know about bone marrow transplants or stem cell transplants. That's really another form of immunotherapy. Many patients receive interferon. This alters how your immune cells work inside the body. And of course, even steroids, corticosteroids like prednisolone, which some of you may have been exposed to. These are examples of non-targeted immune therapies. The difference is that these ones are specifically focused just on the cancer cells rather than working in a broader sense to change how the immune system's working. So why are these so exciting? Well, really, what's the promise? What's the benefit? So these can be very highly specific or selective, so just really pinpoint the cancer cells by using proteins or markers on the cell surface, and they really have the potential to just selectively remove the disease-driving cells, even possibly leading to long-term responses or cures. So that's the goal with these agents, rather than just improving symptoms and reducing spleen sizes. So it's a very different approach. What are the challenges? Well, it's actually quite difficult to discover proteins on the surface of cancer cells that aren't expressed on healthy cells. So that's one of the major challenges in the field. Some types of cancer have a particular marker that's not expressed elsewhere, or even if it is expressed, it doesn't really matter. But with cancers of the blood system, it's difficult to distinguish the cancerous blood cells from the healthy ones, and that's what we really need to be able to do. Not all patients' cancers are the same in other settings. And also some of these therapies can overactivate the immune system, which can cause side effects. And also in other settings, for example, in some certain types of leukemias and lymphomas, we know that when we target a cancer by targeting one specific marker, the cancer cell can then get clever and get around that by just losing that marker altogether, and then you evolve a clone or a group of cells which doesn't any longer express that target. So those are the types of challenges, but they have already been successful in the field. So why has this come to the front of our mind now? Well, I think most people in the room will have heard of the JAK2 mutation, the JAK2 V617F, which is one of the key driver mutations that causes a myeloproliferative neoplasm. There's another mutation that's the common mutation that occurs in people who don't have the JAK2 mutation, and that affects a protein called calreticulin. And this might look a bit complicated, but I'm going to walk you through it now. So calreticulin is a protein that's normally present in all of the cells in our body, and it plays an important role. It's called a chaperone, and that really means it guides proteins and guides other molecules, telling them where to go inside the cell. Now, as we're getting older, we often get little typing errors in our genetic code, and that changes how the genes are read and how proteins are made inside our body. And when you get a mutation in the gene that's encoding for calreticulin, that changes how this protein works. It loses the signal that tells it to stay put inside the middle of the cell and keep away from the cell surface. But it still binds to the proteins that it chaperones. And one of the proteins that it binds is this protein that's actually a receptor for a growth factor or a signal coming from the outside. So you get this complex forming inside the cell, and it travels up, and then it sits on the surface of the cell. And the cell I'm showing here, this is depicting what's called a blood stem cell. So we will have these cells inside our bone marrow, and those are the cells that produce all the circulating blood cells inside our body throughout our lives. So normally those cells are sitting quiet and they're waiting for signals telling them how many cells to make. But when a cell acquires this mutation, it expresses the calreticulin mutation, or indeed has a mutation affecting JAK2, which you can see here, it's no longer listening for signals. So now it's just pumping out cells, as many as it likes, without listening for those signals, and you get this dysregulation of the number of cells that are circulating in the blood and that are present inside the bone marrow. So what's particularly relevant for this talk is you now have a situation where this mutant protein that's only present in the cancer cells is robustly displayed on the surface of the cancer cells. And this is actually a really unique opportunity to specifically target the cancer cell using then antibodies or other immune therapies that can target those cells. So that's why it's quite exciting. And really, it's the only example actually in cancer biology where you have what's called a cancer neoantigen, so a completely new protein produced by a cancer cell that's expressed there on the cell surface. So there's a lot of excitement and interest to try and now target that with immune therapies such as antibodies. So there are 3 trials which are coming on the horizon. There's a vaccine study which opened a couple of months ago, and this is the study where we're about to start enrolling patients now in the UK. There have been about 10 to 15 patients enrolled worldwide, and there are 2 trials of therapeutic antibodies, one of which has opened in other sites internationally and will open here in the UK probably, I think, January to February, and then a second antibody trial which will open slightly later in the year. This one was presented actually in what's called the plenary at ASH. So every Christmas you get, it's about 40,000 haematologists internationally gather in a city in the US. So we have a very big meeting. And then the most exciting news stories are presented to the group. So at that plenary last year, one of these antibodies was presented, really showing how exciting it is for the field. And then this second antibody will be presented at the same meeting this year. So I'm going to start with the vaccine study. So this doesn't involve antibodies. What this is basically giving patients a vaccine. And how does the vaccine work? Well, it's a bit like sending your immune cells to the gym. So it's giving them repeated exposures to the protein that they need to recognize and target, and arming them, causing them to build up their ability to recognize and clear that from the system. So these trials have actually been done, trials of vaccines have been done before. There was a trial published a couple of years ago, and what that trial told us was it was possible, basically, to arm and train the immune system to recognize the mutant calreticulin protein. So what I'm showing in these plots here, these are T cells in this dish from a patient who didn't receive a vaccine, before they received the vaccine, and here's the T cells from the patient at the beginning, after 3 vaccines, 6 vaccines, 15 vaccines. And each spot here on this plot is showing you that the T cells recognize the mutant calreticulin protein, so now it's causing a response. So it really showed us that it was possible to reawaken patients' T cells to attack the mutant calreticulin protein if you give it enough training. That particular trial didn't really show there were any clinical responses, but there are many reasons why that could happen, and that's what's been improved on in the design of this next trial. So why don't the body's T cells clear the cancer cells in the first place? I've got a couple of cartoons that I found on the internet from a university in the US that I thought really explained this very nicely. So what does the immune system need in order to recognize a protein that shouldn't be there? Well, you have these cells that are called antigen-presenting cells. That's depicted in this purple cell here, and that basically gathers pieces of protein that shouldn't be there and presents it then to cells called T cells in your body that are the attacking cells. That's why they're shown with their bows and arrows. So sometimes these antigen-presenting cells, as they're called, they're not doing this job very effectively. And there was a really nice study last year that showed that in some people, for some reason, their antigen-presenting cells are not particularly good at presenting antigens. They sort of wave them around, but don't specifically give them to the T cells. And that means the T cells don't bother sort of coming into action. When that does happen, so if they correctly meet and they pass over the message, they're basically giving them a big go system, a go signal, and the T cells should then do their attacking like this. So these now are the cancer cells. The T cells should do their business and attack the cancer cells. Now, what can also happen, sometimes those cancer cells, they get a bit clever and they express proteins, other proteins, that basically knock out the T cells and send them to sleep. And this, actually, there was the Nobel Prize was given for the discovery of this interaction a couple of years ago. So one of the reasons why these vaccines didn't work properly in the past was because we know that people with MPNs, from other scientific research that's been done, we know that the people who go on to develop an MPN, we know that their T cells are not very good at recognizing the MPN calreticulin proteins, the mutant calreticulin or the mutant JAK2. And actually what we also know is when we study healthy people without an MPN, some of those people actually have T cells that have previously seen the mutant calreticulin protein and cleared it. So we know that the people who don't develop an MPN are able to clear it, but those that do, their T cells and their antigen-presenting cells aren't working effectively, and that's why the MPN clone is able to grow out. So the vaccine trial that's coming now, it's using a vaccine that's basically very similar really to the COVID vaccine. It's given over a course of 9 visits, over 11 visits in total, and you get a combination of a boost and a prime vaccine. So they've formulated 2 different types of vaccine, and the idea is a bit like when we were in the COVID vaccine era, people were being told if you had the AstraZeneca vaccine and then you had Moderna, you got better protection. The same approach is being used here where you get multiple different types of vaccine to really train the immune system to recognize that protein. And then every time you get a dose of the vaccine, you also get a drug called ipilimumab that's basically blocking that sleeping signal. So it's blocking the stop signal that some of the cancers express that causes the T cells not to act. So this is quite a new approach and hasn't been given in this way before. And the idea is we hope that by using these different techniques of an improved vaccine that's presented better on those antigen-presenting cells and reawakening the T cells, the idea is that your own T cells would then be able to act like they do in a healthy person and clear the MPN away. So that's the first study. In terms of the actual involvement in the study, it's relatively light touch. I thought Margaret spoke really nicely about the sort of level of engagement that's required to be on a clinical trial with multiple visits. This one is relatively light touch, so it's fewer visits spread out, and I think each of those individual visits is a little bit less. So then coming on to the antibody trials, so these are of 2 different types, and I'm just going to explain briefly why they're so different. The first trial is an antibody that basically blocks that signal, the signal that's always on. So I talked about how these mutations stop those stem cells they stop the stem cells for waiting for signals, telling them how many cells to produce. They're always just pumping out, churning out cells. What this antibody does is it sits there in the pocket of the receptor and it blocks it and tells it, actually, guys, just shut up, stop signaling, stop doing what you're doing. And in a way, this is a little bit similar to a JAK inhibitor, but it's only acting on the mutant calreticulin protein, so it's working by a similar way. It's inhibiting that signaling, but only doing it on the cells that carry the mutant calreticulin protein. The way it's doing it is quite interesting, and just in case you want to know, so basically that receptor that's listening for signals, you need 2 halves of it, so each half is expressed separately inside a cell, and then they come together here at the cell surface. That's 1 half, and here's another half. The antibody sits in that pocket and it pushes those 2 halves away, and that stops the signaling happening. So that's how it's working. And this has been really beautifully shown by a number of collaborators across the UK and throughout the US as well, who collaborated with the drug company to develop the antibody. And I'm just really mentioning that because it's really important, I think, to recognise how pharmaceutical companies actually work with academic groups in order to figure out the right approach. And just really one slide on what they presented last year. So we often look at these conditions in vivo, in mouse models. So this is a picture of the bone marrow from a mouse who's got an MPN. And you can see all these dark cells here. These are the platelet-producing cells, the megakaryocytes, that shouldn't really be there in that high number. So this mouse has a very, very high platelet count. This is a mouse who received no treatment. This is a mouse who received this therapeutic antibody, and you can see the very dramatic clearance of those cells. So this is ideally what we're hoping to then achieve in patients. Now, the second antibody trial is really quite different. So it's not targeting that inhibition. Instead, what it's doing, here's the antibody here in this picture, there's the cancer cell and there's the T cell. So it sits here, binds to the cancer cell, and then it has another arm, and it basically, the other arm grabs the T cells as they go past, and it says, hey guys, come and look, you need to kill this. And then the T cell releases the products that then cause that cancer cell to break up and go away. So this is what's called a bispecific antibody. It's bispecific because it's got those two arms, one binding the cancer cell, one binding the T cell, and then the idea is that you then use the body's own T cells to clear the cancer. So it acts as a bridge between the cancer cell and the T cell. We know it recognises all the different types of the calreticulin mutations, and what they're going to tell us this year at ASH is that they've seen very effectively that it kills MPN cells from patients when you put them in a dish. And also they've done some work in preclinical models, as in mouse models, and apparently it works very well there too. We've not seen the data, but we're looking forward to seeing that in December. So this study will open towards the end of next year. So what are the challenges and uncertainties? Obviously, I've told you a lot about the promise. So will these treatment work actually on the cancer stem cells? In the picture I showed you, where I showed you clearance of those dark brown cells, those are the platelet-producing cells. So those platelet-producing cells, they're important in driving the inflammation and the fibrosis, but they're not the cells that actually start the cancer. The cells that start the cancer are called stem cells. They also express the mutant calreticulin, but we don't know how much actually. They express the mutant calreticulin protein a little bit less than the platelet-producing cells. So one of the challenges will be to see how well these therapies clear those underlying stem cells. And then the second big challenge, Adam was talking about inflammation. We also know that many patients have fibrosis in the bone marrow, those who've got myelofibrosis. We actually don't know how well T cells will work in this kind of scarred and inflamed bone marrow microenvironment. So that's another challenge. And just to talk a little bit more about the science that we also do, one of the other techniques that we use in our labs in Oxford to understand these cancers is we actually grow little bone marrows in a dish from stem cells from healthy people. So we're able to get these stem cells to basically generate hundreds of little bone marrows in a dish, and we can then take cells from patients. These green cells here are cells from patients that we then seed into these bone marrows to look to see how they alter the bone marrow microenvironment and also how that microenvironment changes how the patient's cells work. So one of the things we can do then is cause these little they're called mini organoids. We can cause them to become fibrotic and then see how these antibodies might work within a healthy bone marrow or within a fibrotic bone marrow. And this is an example of some of the scientists who really are doing all this work here in the labs. And this is what we'll do then. So we'll have mini organs that are healthy and then organoids where we've caused them to become fibrotic and inflamed, and see how a change to this microenvironment will alter how these antibodies might be effective. So we've talked about the promise here, and just to summarise then, I've talked you through vaccines and two different types of antibody therapies. So the vaccine trial is training the immune system and waking up the T cells, and the two types of therapeutic antibodies. One is trying to turn off this switch that's otherwise always on, this inhibitory antibody, and the second is bringing in T cells to kill the mutant calreticulin positive cells. And one thing just to highlight, which maybe I didn't mention it clearly enough before, the vaccine trial is open not just to patients with mutant calreticulin, but also to patients who've got a JAK2 mutation, but only to those with a particular blood cell type. All of the trials obviously have quite specific eligibility criteria. And then finally, just to add my voice as well, to say thank you to all for donating samples to the research that really discovers these types of therapies, participating in trials, and also to patients who play a really important role in informing our research by participating in some of the priority research setting workshops that we do. And in particular, again, to thank the funders who fund our careers, much of the science that goes on, and enables days like today to happen. So thank you very much.